Starting with carbohydrates, these are essential biomolecules found abundantly in our diet. To confirm their presence, scientists employ several intriguing tests. The first is the Molecules test, where a sample of food is mixed with Molecules reagent. If a purple or violet ring forms at the interface, it confirms the presence of carbohydrates. Next, we have the Fehling's test and Benedict's test. Both these tests involve adding specific reagents to the food sample. The appearance of a red precipitate in either test is a clear indicator that carbohydrates are present. But not all carbohydrates react the same way. For instance, in the Tollens test, a food sample treated with Tollens reagent results in a silver mirror formation, particularly indicating reducing sugars. Another specific carbohydrate test is the iodine test. When iodine solution is added to a food sample, a blue coloration indicates the presence of starch, a complex carbohydrate. Moving on to proteins, these are the building blocks of life, vital for various bodily functions. To detect proteins, the Biorate test is commonly used. Here, aqueous copper sulfate is added to the food sample. A violet coloration confirms the presence of proteins. The xanthoproteic test involves treating the food sample with nitric acid. A yellow color solution indicates that proteins are indeed present. Similarly, the Millions test uses mercuric sulfate in the presence of sodium nitrite and sulfuric acid to produce a brick red color solution, again confirming proteins. Another method is the ninhydrin test, where a pyridine solution of ninhydrin is added to the sample. The formation of a violet color solution is a positive indication of the presence of proteins. Lastly, let's talk about fats, which are crucial for storing energy. The solubility test is straightforward. A food sample is mixed with chloroform or alcohol. If it becomes miscible with chloroform and immiscible with water, this confirms the presence of fats. The translucent spot test is another simple method. A food sample is rubbed between the folds of filter paper. The appearance of a translucent spot indicates the presence of fats. Finally, the acrolein test involves heating the food sample with potassium bisulfite. The emission of a pungent, irritating odor confirms the presence of fats or oils. To summarize, detecting carbohydrates, proteins, and fats in food involves a variety of fascinating chemical reactions. From the color changes in the Biorate and xanthoproteic tests for proteins to the silver mirror formation in the Tollens test for carbohydrates, each method provides crucial information about what's in our food. Understanding these tests not only deepens our appreciation for the science behind nutrition, but also enhances our awareness of the nutrients we consume daily. So the next time you take a bite, remember the intricate dance of molecules that were confirmed by these amazing scientific tests.